Have you ever wondered what happens to the water you use after it circles the drain? Microorganisms remove pollutants and pathogens from water during wastewater treatment. Without them, we would be drowning in poop and all of the diseases that go along with it. Microorganisms actually help us do many things. They help us make cheese and yogurt. They make crops grow stronger. They make chemicals that we use in many different industries, and they're the reason we even have antibiotics and certain vaccines. Studying microbial life in the laboratory has given us groundbreaking discoveries. But scientists estimate that we're only able to grow 2% of microbes on Earth in the lab. Just 2%. 2% has given us those antibiotics. 2% has given us the vaccine for malaria. And we're grateful for the 2%, don't get me wrong. But we live in a time where antibiotic resistance is a greater problem than ever before. Water security threatens the most developed countries, and we need more than just 2%. So I'm going after the other 98%. Let me tell you how. The past few decades of research have given us an explosion in the amount of data we have on microbial communities, starting with the thousands of genes encoded in a single organism's DNA, all the way through to the chemicals they're actually producing and consuming as a community. I'm talking big data. But what we don't yet have is an efficient way to use that big data. I'm using artificial intelligence and machine learning to mine the big data from real-world microbial communities living outside of the lab in their own environment. With the power of computing, I can draw connections among millions of data points that we as researchers might miss if we were looking at the data ourselves. Wait a second, that sounds cool, but what does it really mean? This big data we're talking about has a few different forms. The data that can tell us about every gene in a single organism's DNA looks like this. A long string of A's, T's, G's, and C's. That's basically a code for what the organism is genetically programmed to be able to do. Other big data looks really similar in the computer, but can tell us which of these genes were actually being used by the organism at a certain time. We get this data from a microbe's RNA. Another big data set we work with looks much different and is processed in a very different way. This data set tells us what compounds and nutrients were found inside or outside of the cells at a certain time. We call this data the metabolites. When we put this all together, we get a nice picture of what the cells can do, what they are doing, and what's happening in and around them as a result. And that's only one full set of data. If we look at many, many data sets, the computer can learn to recognize patterns and draw connections from new data based on data it has already seen. And that's just one way we can apply machine learning. Using this approach, we can determine the exact conditions that these microbes need to thrive, mimic those conditions back at the lab, and increase that 2%. Imagine what the other 98% could give us. The next antibiotic to keep us ahead of resistance. An organism that could mineralize salt out of seawater for on-demand desalination anytime, any place. An organism that could remove dangerous pathogens from indoor air. The undiscovered 98% will shape our world in ways we have yet to imagine.